My name is Lichtenstein. For those of you who know me, I'm sorry. And for those of you who don't know me, well, I'm sorry in advance. So today, I'm going to be talking about somatic variant discovery. And if you think to yourself, well, what's somatic variant discovery? Don't worry, it's in the slides. So we're going to be talking kind of about an overview of somatic variant discovery, as well as a bit about the workflows themselves to do somatic variant discovery in the GATK. So I want to talk briefly about mutation events and tumor progression. In case you did not already know, when we say somatic events, most of the time we are talking about cancer. So the main point that I want to kind of illustrate with this slide is when you take a sample, you're cutting out a bunch of cells. The mutation profile is actually very heterogeneous. So it's not like calling germline mutations. You know, in other words, it's harder. Don't tell anyone from the germline side I said that. If you take time, you start with some kind of event, some kind of mutations get introduced, and that is your clonal mutation, parentheses S. And what that does is it kicks off a population of malignant cells, and then you start getting other events. Some help the subpopulation survive. That's the big red lightning bolts. And then some just kind of are along for the ride. But when you do the sequencing, you're going to see kind of at the time the events that are in all of the populations. So in order to, unlike germline variation, in order to detect driver mutations, your germline variation is kind of what's in your normal sample. So typically this would be a blood sample. In fact, I recommend using blood samples. And then you also have somatic alterations. These are typically what would be in the tumor. So a somatic alteration would be only in one subpopulation of your cells. So in germline, you often have these large cohorts to help to help give you statistical power. In cancer, typically, if you are presented with one tumor normal pair, you would use other studies to determine, well, is this a common mutation that I see in cancer? So let me go back and give a little bit of terminology for the tumor heterogeneity. So when I talk about purity, I'm talking about the proportion of the cells in the tumor that are actually tumor cells. You can think that when someone goes in and cuts a biopsy of a tumor, they're also going to get normal cells with it, i.e. ones with only germline mutations. So we use the purity to indicate in this sample what is the proportion of cells that are from the actual tumor. And then tumor heterogeneity is based on the idea that you have all those different subpopulations, as I explained on a previous slide. So what does this look like in a sequencer? I don't know how many of you are familiar with calling variants from sequencing data, but I figure you want to learn, and that's why you're here. So there's actually a breadth of variants. A lot of the time people say, well, I do mutation calling, but all they really mean is they're doing germline, SNPs, and indels. So let's start. What's a SNP and an indel? Well, at the top, you have a reference sequence. And essentially, when you're calling variants, you're looking for differences between your sample and the reference sequence. So a point mutation is one base got changed. I'd love it. Is there a laser pointer? OK, thanks. One base got changed. So in this case, a C got changed to an A. And what that will look like is in the reads, at that location, you will see that the reads basically, at that position, all say C when the reference says A. Indels are small insertions or deletions of bases. So what you'll see again is that the reads are a little bit broken. If it's a deletion or if it's an insertion, they'll look a little longer. Those are two kinds of variants. There's also other classes of variants, such as copy number alterations. In that case, what you have is a piece of the genome has essentially been duplicated or removed relative to the reference. So what you end up seeing is either too few reads than you would expect, either half or zero typically, or if you have a duplication, you'll see extra reads relative to what you would expect, because now more DNA represents that region, so you'll get more reads. In the case of what we'll be talking about today, we're mostly going to just be focusing on the small mutations, which are SNVs and indels, and the copy number alterations, which are deletions and amplifications, sometimes also called gains. There's also structural variants and pathogen detection, which I will not be talking about in this presentation, but for your edification, I will go over briefly. Structural variants are a wide class of variants, including copy number alterations, but we actually tease them apart for the purposes of explaining the tools. So structural variant would also include like a translocation breakpoint where, say, essentially like a piece of chromosome 5 has glued itself to chromosome 1. So when you do the sequencing of the read, it's like you get reads that look like chromosome 1, chromosome 1, chromosome 1, chromosome 5, chromosome 5, chromosome 5, chromosome 5. And so that would be a translocation, and there are tools for doing that. We also can detect pathogens. So some pathogens work by kind of altering the sequence, inserting sequence into the DNA. And in those cases, we do have a tool for that called PathSeq, which again, I will not be explaining today. And in that case, what we do is we look for reads that align to a completely different reference. Now, for all of these variants, these variant classes, you can have germline, which is basically stuff that you're born with. Basically, they're in all of your cells relative to the reference. Or you can have somatic, and somatic means, again, just a subpopulation of the cells in your body have the variants. So essentially, how the tools in the GATK tend to be broken up are, you don't have a copy number tool, you will have a germline copy number tool. Then you will also have a somatic copy number tool, or you will have a germline SNP and indel caller, or you will have a somatic SNP and indel caller. These are all going to be different tools. And today I'm going to only be explaining the somatic ones, or giving you an overview of the somatic ones. So I like to always tease the germline people about how easy their job is compared to those of us on the somatic side. And if any of you are doing germline analysis, I don't know what to tell you. So here is why I say it. Here is why I say it. It's because when I have a germline variant, essentially, let's just take a heterozygous SNP, I would expect about 50% of the reads to support the alternate allele. 50%. If there are 100 reads there, you've got 50 of them. Like, you should be able to do that. That's not hard. Whereas when I have a somatic variant, there's like heterogeneity, which dilute the number of reads. There's contamination, both from cross-individual or from the normal. There's other mutations that are in there. Maybe you have a little mosaicism. By and large, I can't rely on the ploidy anymore. Like for germline calling, I can say, well, I have 2 strands. If one of them is different, I expect half of the reads. Here, I can't rely on that. It won't work. I mean, let's take this example. I have 1 mutation here. That means 50% of the reads will support the alternate allele. In the somatic land, okay, so we took a biopsy. It turned out we had a purity of 67%. 33% of them have no changes at all. But then we have 2 subpopulations in the tumor. One of them has only mutation A. The other has mutation A and mutation B. That means I only have 20% of the reads to support mutation A and 10% of the reads to support mutation B. So now when I used to have 50 reads supporting my mutation, now I only have 10. See, life's hard. And then, because of this, on the germline side, there are artifacts that they don't have to worry about because they affect a small number of reads. So typically, if you're looking for these kind of high percentage of the reads supporting an allele, you don't have to worry because these artifacts or other issues only affect a small number of the reads. But when you're doing somatic calling, If you have like 5 reads out of 100 or 3 reads out of 100, you may want to call that. And you need to start worrying about things like sequencing artifacts and cross-individual contamination, because now suddenly I have someone else's germline variants in 1% of my data, and suddenly I'm going to start saying, oh, look, I'm calling these mutations that are just at a very low allele fraction. But in reality, you're just calling contamination. And of course, there is, other mutation types, the copy number, which can further complicate this, because if I have a duplication, the variant may or may not be in that duplication. So now, again, it's like yet another confound for me to determine how many reads I have to support a variant. Apologies to all the germline people. So again, I'm not going to go over this slide in much depth, but on the somatic side, to do cancer analysis, we have to take into account a lot of confounds that either don't exist in the germline world, or because you're looking for a fixed ploidy of 2, maybe 3, maybe 4, you aren't going to be statistically relevant. And somatic alterations can be dramatic. Here we see a normal cell. You've got your 22 chromosomes. In this case, you have 2 X chromosomes. And then looking at the cancer cell line HCC 1954, I just don't even want to get into this. This thing is just a mess. But this is an actual sample, and I can also show you, non-cell line samples that have similar amounts of mutations. All right, so I've now given you the broad overview. I hope I'm not racing through the slides too fast. By the way, people should feel free to interrupt me and ask me a question. Otherwise, I'm just going to go through the slides. So in GATK4, we have 2 pipelines for calling variants. The first is our small mutation, which is a SNV and indel. And as you'll see, it is based on the tool Mutec2. I will get into that momentarily. We also have somatic CNV calling, and that is based on a tool called model segments. Both of these, this is the kind of schematic of what each tool is doing, or in fact, these are actually, you may want to view them as a sequence of tools. None of these are one command line. These are all multiple command lines that have to be run in a particular order. And as you'll be introduced tomorrow, we actually offer kind of essentially WDL, which is a way of packaging the entire workflow. So you don't have to write like a bash script that's going to say, okay, first I have to call the proportional coverage, and then I have to call normalization, and then I have to call the segmenter. We kind of will just give it to you in a nice handy package. And there are advantages to using WDL and Cromwell over writing a bash script. So I will go now into a little more depth of what these workflows are doing. So let's start with somatic SNVs and indels. So here's the logic of a tumor normal workflow. Now, just to be clear, because I don't know that this is in the slides, you can do tumor-only calling for small variants, but there are disadvantages to do that. You will have a higher false positive rate. I can pretty much guarantee that. But if you don't have a matched normal, I would recommend doing tumor-only. Anyway, so here's how we generally do, at 10,000 feet, how we generally do small variant calling for somatic small mutations. First things first, you don't want to call germline, right? So not only do the germline people get to make fun of me because they can hit deadlines, they also ruin my data. So the germline SNP, we'll say, is right here. You can see roughly 50% of the reads have the SNP, but it's also going to be in my tumor because it's the normal. I mean, the tumor probably isn't going to delete this. I mean, it can, but I'll get into that later for people who want to ask after class. And here is probably a somatic SNV because if you look, there's nothing matching in the normal. So generally speaking, assuming that this passes all the artifact checks and the various filters we have, we would want to call this, but we would not want to call this. Is everyone with me? All right, great. I'm getting nodding heads, bam. Okay. Oh, look at that, there is a slide for that. Okay, So it is possible to run the workflow without a matched normal in tumor-only mode. In fact, we do that when we create panels of normals. For those of you who don't know, a panel of normals is we essentially call a bunch of normals as if they were tumors in tumor-only mode, and that gives us an idea of germline variants and noise that the sequencer likes to make in certain regions of the genomes. Then we kind of boil that down, and we say, okay, like this is a panel of normals. This is kind of the mutations we would expect to get erroneously. And then we use that panel of normals as an input to the actual caller to say, regardless of what's in the matched normal, beware of the stuff that's in here. We don't simply eliminate it. I'm not going to go into that algorithm. But we do kind of, when we model the probability, we kind of lower it to say, well, we saw it in the panel of normals, so it's less likely to be real. So if you're calling in tumor only, it is important to have a good PON. If you don't have a good PON, what's going to happen is you're going to start calling sequencer errors and germline variants as somatic variants. Oh, questions, awesome. You.
I'm not sure we're going to talk about that in the future, but will you go over discovering somatic variants from RNA-seq data? Is the flow pretty similar, the workflow for that? Like, say, for like...
Okay, so if I zoom out enough, I can answer that question with yes. I don't know that in this workshop there's any talk of calling somatic variants in RNA. I have seen, it has definitely been done with, at least, the last time I was involved in it was a long time ago. I don't want to give that away. You'll think I'm old. And we were using the predecessor tool of Mutate2, Mutate1, and we were able to call RNA with it. I would imagine, though, you're not going to be able to just grab the workflow and do it. You'll probably have to change some parameters. But that's why, on the GATK side, we have a forum you can ask, and the developer of Mutate2 in particular is a big fan of answering of helping users. I am too, but you're going to get him before me. The other thing to keep in mind is copy number variants. You're not going to call that through RNA.
What about good on? I mean, well, in a situation where, for example, you don't have a sample run on the same machine, it's better to keep an external PON, go without PON. What is
okay, so the one thing we like to throw out, we use a PON, but actually I have to tell you, one of the things we'd like to be developing is a way to go Pondless, because both the copy number and small mutations use PONs. Because logistically, they're actually difficult and annoying. Because it kind of implies like, I want to start calling my data, but I need to have already sequenced anywhere from like 3 to 150 normals to build up a reasonable PON. So they are a logistic nightmare. And yes, we aren't going to talk too much about that other than maybe a slide to say they are logistically difficult. It is like when we are setting up to do a new study, something like that, or we've changed a sequencing protocol, that is always the most, oh wow, thank you. That is always the most kind of irritating part because I'm telling the sequencing platform, like, I need normals to build up these PONs, otherwise the tools aren't going to perform anywhere near as well.
Is there upfront assessment of the germline, of how good that germline is? Because if we're taking germline from the cheek, then you're likely, okay, fine, that's going to be germline. But then if it's a tumor normal from, say, kidney tissue where you might have infiltration of tumor.
So here is, okay, so that's why originally I was saying if you have a matched normal, try to use blood. There may not be a slide here. I know there's one probably later in the presentation about like, what are good samples for a PON? And obviously having tumor infiltration of the normal, that would be bad to put into your PON because you're essentially saying that is a normal. So there are ways to detect when that is happening. I would have to double check because there have been many iterations of like kind of bad PON detection, and I'm not quite sure where the standard is right now. Okay, but yeah, that is, when I talk about the logistics problem, that's part of it.
Dwelling into the same thing. So let's say if I have a sample and a matched normal, does the panel of normal exclude that matched normal?
No. So they're used tangential. So for instance, if I see sorry, excuse me. So if I see, what is happening? Okay, so if I see a variant in the matched normal, I'm going to weight that more heavily than I saw it in the pond, but not in the matched normal. Okay? Essentially, but they are both used. And you can run without a pond, but you run the risk of kind of getting sequencer errors because they might not be in the normal, but they just happen to be in the tumor. Many people also sequence the tumor to a deeper depth, which of course means you might have like a few as opposed to 0. So we recommend both, and obviously matching your kind of, as he was getting at, matching your protocol of how you generated the samples for the case samples and for the normal, maybe with the exception of depth, like we can deal with differential depth in these tools. Anything else? Excellent. I might blow through this half hour. Okay. So essentially, the PON is just a VCF made from a set of unrelated normal samples. As I said already, the purpose of it is to eliminate common and recurring technical. The secondary purpose is also to eliminate germline variants or to kind of approximate a normal if you don't have a normal available. Okay, I'm going to move on to copy number variation unless someone stops me and wants to ask another question about small mutations. Going once, going twice, gone. Okay, so here is an overview of the workflow for somatic CNV. Essentially, you take your reads and you kind of cut up the genome either into the targets if you're doing whole exome or some kind of capture kit, or you bin the whole genome. You collect your proportional coverage, and that's going to kind of tell you what you have in your sample, and then you normalize it against a panel of normals. As you can see, that's one of the inputs. You segment it, you get a coverage profile, you get to plot it. I'll talk about calling here as well. And then your output is basically segments where you can say what the copy ratio is for a particular segment and an estimation of whether it believes it's copy neutral, deleted, or amplified. And you get plots. So let's start first about collecting the proportional coverage. So let's say this bar is your genome. Your target is this dark gray. Essentially, all you're doing is you're just counting the reads who overlap this bar. That's it. And you get a big file where all your bins and all of your targets have some kind of count. Then what you do is you normalize it. So if I had 100 of reads total, this bin had 50 reads, the proportional coverage would be 0.5. Then what we do is we take the PON, and the PON has two key pieces of information in it. The first, which is probably the easiest, is of all the normals that I put in the PON, what was the median proportional coverage I would expect at this location? And then that gets turned into essentially a ratio, such as if I had slightly more, it might be 1.1 now. And then we do, I'll get to it in a moment, we'll do a denoising step to essentially turn thick clouds of estimates and make the estimates tighter. I do think we will be talking about that in here. And then eventually what we're going to do is draw segments. That's what these black lines are, where we say this is a contiguous region of the genome where we believe the copy ratio is the same, and in this case that copy ratio is 1. For this segment right here, it's about 1. Just so you know, the blue versus orange, we're just rotating the colors. There's no difference between blue and orange. It's just so that you can see where there are breakpoints. So here comes the denoising. You have raw copy ratio, raw counts. Like, this is not useful. If any of you can see stuff in here, like, hats off to you. You are a psychic. Then we do kind of the normalization against the median. And as you can see, like, you're starting to see some shape, but it's still pretty noisy. Then we do the denoising step, which I don't know that we talk about in these slides, but if anyone wants, I can happily explain it to them later, which, as you can see, in order to get better segmentation, we want this width to shrink. And as you can see, that's exactly what happens. Then taking these points, which were basically just points of proportional coverage that we're transforming a bunch of times, we then do the segmentation, and that's when we draw the lines through it. Then we have a caller, which given the information, we say, okay, like if it's in one particular region, we say, okay, it wasn't, there's no change here. It's what we would expect. It's copy neutral, meaning if it's a human, it's probably just got 2 copies here. But for a segment like this, which is shifted down, although sorry, there's no axes here, we would say this is probably a deletion. And these that are shifted up are probably amplifications. Any questions? Because I just dumped the load on. Yes?
When you go from blue to green, can you reiterate what happened?
Okay. Do you want I will try to give you the quick version. Essentially what's happening here is we now have a copy ratio estimate that is essentially just units of median, units of the median in the pond. What we do before, when we created the pond, we basically did an SVD to capture a hyperplane using the normal samples. You're with me so far? Now, you can take your case sample and you project it into that hyperplane. And what you're saying, there's a nice, fast linear algebra trick to do that. And what it does is it says this is kind of, from the case sample to this projection, is the least error in the hyperplane. Now, by the fact that you're in the hyperplane, intuitively what you can say is this projection is the closest thing to a normal that this case sample, for this case sample. So then what we do is we just subtract the case sample from the projection, and it kind of pulls out all of the systematic noise that you would have seen if you were sequencing a normal. And that's how this shrinks, and since there's no good linear combination that will get this back to 0, it stays out here. But there's some structure in here, you can't see it with your naked eye, that is getting removed because essentially you would see it in the normals as well if you sequenced it. So, you know, something, although we do, I don't talk about it here, we do explicitly model GC content. GC content used to be something we would see get removed a lot from this process. Any other questions? Okay, I'm going to move on then, because I'm in danger of running over. So the panel of normals from the CNV, I have to emphasize, is not the same file as the panel of normals you use for the small mutations. I'm sorry, it's a little confusing, but that's how it is. So it is not a VCF. It is an HDF5 file with a bunch of matrices and metadata about the samples you used for your panel of normals. Again, as I was talking about, its main purpose is basically to denoise your initial copy ratio estimates. Again, I don't go into a ton of detail here, but the normals should be the same protocol used for sequencing. The normals should be what you're going to use for your case sample as well. I would say the difference is the depth of coverage tends not to be a big thing. In other words, don't worry if the normals were sequenced at a different coverage than the tumors. This procedure will still work. But I will definitely say the more coverage, the better, always. Yes?
If you had tumor normal pairs, can you take all the normals and make your pond?
Yeah, people do that. You could do that if you were going to run a cohort of like, I don't know, 20 to 200, whatever it is. A lot of the time what people will do is they will just take the normals that they're using in their paired and make a pond. And then they can use that for the CNV calling. Just as a warning, we also do, as part of the pipeline, we also do the copy number calling of the normal sample. If the normal is in the pond, you are going to get basically straight lines. So don't use that data because it's not worthwhile. But that's from the vast majority of use cases, that is a very small caveat that no one typically cares about.
And what about adjacent normals? In like a cancer setting, they're many times considered not real normals, but they're not the tumor.
So you can use them, but you may see more. What you might see depending on how bad it is. So first of all, let me just say I would highly recommend using blood normals. Not only that, I would even recommend use blood normals sequenced the same way from different individuals, if you can. Again, when I keep talking about the logistics headaches, there's this. And because you can still run into issues where if you do have somatic variants in the panel, it will attenuate the real somatic variants that match that in the case samples. So usually, if I have a bunch of matched normals, usually they're not majority tissue normals, at least with the data I've been working with. If that's not the case for you, I'm sorry. This hasn't been a huge issue typically, but we have seen it. And also, you typically want patients maybe who, or at least the people who are populating your panel of normals, to be kind of on the younger side, because you can get germline mosaicism that can also confound this.
And if you did have, like, say, a panel of normals from blood normals, and you had adjacent normal tissue, you can call copy number, you're saying?
Oh, you're talking about calling it. I thought you were talking about using the.
Initially, I was talking
Oh, okay.
about using it.
Yeah. As long the key piece is if the sample is in the panel, then you're going to just get a straight line, because it's basically just going to find a linear combination with itself and say, okay, here you go. And the SVD will make it so that there's like this tiny bit of normal. If I had, I could show you samples where I did this versus when I used a pond that didn't, but just, you can see it immediately because your noise is whisper thin. Oh, wow, yeah.
Can you use also for target sequencing or only?
Okay, the answer is yes. You can, we don't have any evaluation data on it because every targeting is different. How many targets are you using? I will say if you're using very few targets, you may not want to, pro tip for people out there, if you're using very few targets, what that is, I'm gonna kind of shrug my shoulders, but you may want to just use the estimates without the segmentation, because if you have huge distances between the segments, they can get thrown off. That is not something we've corrected. And as a warning, when we do evaluations, we typically only do it on WGS and whole exome. There are just too many targeted sequencing. There are just too many out there for us to kind of offer that. And some of them are also, clinical samples and things like that. But yeah, the answer is people have done it, and they have gotten results that they liked, but I'm only saying that anecdotally. You had a question.
So just the question before about ponds and using adjacent tissue, this is, I don't understand, is this adjacent tissue to the corresponding tumor? Because that wouldn't be for a pond, right?
Correct. I'm saying you shouldn't use adjacent tissue.
But that would be just a germline match of the same individual. That wouldn't be the case of a pond, because a pond is when we don't have corresponding germline for the same patient, right?
Are we talking about copy number or?
Both.
Okay. I would argue that if you can get a matched normal, always do that. Always do that for small mutation calling. Don't put it in the pond if you can get away with that. Don't put adjacent tissue in the pond.
Do we need the pond if we have a matched germline for the same patient?
I would recommend it to still use it. Your results will definitely improve. But if you had to choose one, I would say the matched normal is definitely more effective. But honestly, especially because of sequencer error, that's really what the pond is actually going to get. We're now talking about the small mutations. Actually, we're talking about both small mutations and copy number.
So if you had whole genome and whole exome data, which would you use for copy number?
Which would I use?
Is one more preferable than the other for calling copy number?
I would probably, it depends what I'm doing, but almost always I would, let's just, first of all, they're not going to be covered to the same depth. If that's the case, then yeah, WGS, of course. You'll wait longer. So it's a little hard. The reason I'm kind of waffling is, if you run it on WGS per sample, it's going to be more expensive. But you're going to get better resolution of where the breakpoints are, right? Because with whole exome, it can only find breakpoints at kind of the targets, whereas if you've carved up the entire genome. So I would say it kind of depends on what you're trying to do.
I guess let me give you a more specific example.
Sure.
So we have like 60x whole exome and 15x whole genome, which would be yours?
Definitely the 60x because, well, it depends, again, it depends what I'm doing downstream, right? I mean, again, do I need the resolution? When I start hearing numbers like 15x, 10x, I start getting worried because when coverage is getting low, there's a certain amount of noise that just cannot be corrected. And the difference between I have more experience with 60x than 15x and know that 60x can still yield something quite good, whereas I don't have a lot of experience with 15x, and so I would be a little more worried.